A very big hello to my wonderful PU2 champions. I hope all of you are doing absolutely great. Well, today we are going to deal with the chapter Principles of Inheritance and Variation. A super important chapter. Yes, and why is it super important? I call it Bahubali of biology because, my dear children, you get straight 13 marks coming from this chapter, and this number is not a small number. And what is the mark distribution like? You'll get 1 marker, 1 2 marker, and there will be 2 5 marker question. 1 marker, 1 2 marker, and there will be 2 5 marker questions coming from this chapter. Got it? I hope you got it. And my dear children, you need to understand one thing. If you are able to grab these 2 5 marker question, what will happen? You will get straight 10 marks. 2 into 5, 10 marks. So make sure you do that. I'll make sure from my end that you can grab those marks. And also, my dear children, I have put a homework question for all of you guys. Make sure you solve it and let me know in the comment section. Do not worry. It's just an MCQ question. And if at all you're liking this session, hit the subscribe button, because or else you might be missing a lot. Okay, let's start with the model question paper questions of the year 2025-26. I have taken 3 MQPs along with their number. So question number 3 of MQP 1 is the first question that we are going to deal with, and it says progenies with AB and O blood groups are not possible from which of the following mating parents? That means, okay, I have to make sure that the progenies with AB and O blood group are not being born. That means the parental combination should be something different. My dear children, if suppose I want AB blood group, I want AB blood group progeny, in that case I have to make sure that parent 1 should have at least 1 allele for A. Other allele can be anything, right? Similarly, parent 2 is there, it should be having 1 allele for B, then only these two can come together and give me AB blood group. But over here, I do not want AB blood group. So I have to make sure that none of the parents should be having A here and B there. In the opposite side, it should not be A and B. For example, if we consider B option, here A blood group is on parent 1, A allele is there in the parent 1. Now B allele is there in parent 2, both A and B can come together to give me AB. So that means I do not want this, because I do not want progenies with AB and O blood group, right? Similarly over here, I have A here, I have B here, these two can combine together. Similarly, I have A here, I have B here, these two also can combine together, giving me AB blood group. So I do not want this also. What about this option? A is there, A is there. Oh, ma'am, it's not possible. No, this will be the answer. Look over here. No, B is also there. This A and B can also combine together to give me AB, right? So this is also not going to be my option. So what I am left with? I am left with this option. Is this correct? Is this correct? When they are fused, my dear children, when a person having O blood group and AB blood group are fused, won't they give O blood group and AB? Let's check it out. So for a person to have O blood group, O is only found when both the recessive alleles are there. When both the alleles are recessive, only in this condition O blood group will be there. And for AB, 1 allele should be A, another allele should be B. Now suppose this and this crosses, I get IA, IO. Now A is dominant over O, which is why the blood group will be A. This is this, IB and IO. B is dominant over O, and that's how I get B blood group. This and this, again IA, IO. A is dominant over O, I get A blood group. This and this, I get IB and IO. B is dominant over O, giving me what? B blood group. Whatever we do, we get A and B. Whatever we do, we are going to get A blood group and B blood group separately. We do not get AB blood group, we do not even get O blood group. So your answer for this question becomes what? This. So while you are looking for AB, look for O blood group as well. Okay, so over here I gave you an example for AB and then only I told you, but in such condition there might be options where O blood group is also there, so please consider all of that. Okay, chalo, let's go ahead with the next question. Now this is a 2 marker question. As I said, only 1 marker question comes, 1 2 marker and 2 5 markers. So recombinants obtained in both Mendelian and Morgan experiment, yet the processes involved are different. Comment on it. So recombinants, what do you mean by that? For example, I fused a green and okay, not green. I fused round and yellow plant with round and yellow seeded plant with wrinkled and green. Wrinkled and green. So in the first generation, the dominant trait appeared, that is round and yellow. All of them were round and yellow. In the second generation, I found seeds which had round and green, which had traits like round and green. Instead of yellow, it was green. So that means it's a non-parental character. It's not a character that you see in parents. Wrinkled was with green. Round was never with green, but you see that. Why, my dear children? Because new combinations have been formed there. Recombinants are coming up there. That is what you mean by recombinants. New combinations are showing up there. Now, there was a person who was doing experiments on chromosomal theory of inheritance. His name was Morgan. And Mendel, do you know, Gregor Mendel, he was doing his regular experiment. Both of them got recombinants. They both got recombinants, but by a different mechanism. So they are asking you, what is the difference? You have to comment about that. So my dear children, in Mendel's experiment, I can tell you something very easily. Over here, suppose there are 4 chromosomes. After them, half of them can go in one cell and half can go in another cell. In one, over here, my dear children, suppose the pink and the blue is going. The pink and the blue is going. In another one, the red and the blue is going. Light blue is going. Fine. This happened by chance. Okay, from this pair, from the top pair, one had to go, so this one went. From this pair, one had to go, so this one went. It was by chance. Now over here, from another pair, again, only one can go. So it can be either light blue, but over here it's not light blue, it's dark blue. Okay, that went. But the light blue one can also go. You need to understand that light blue one can also go. So suppose if at all this picture is too much confusing for you, I can give you example by my own picture. You have chromosomes like this. Okay, fine. Now over here, this is a chromosome pair and this is a chromosome pair. Fine. From this category, one can go into a gamete. One will be going into a gamete because in the gamete, half the number of chromosomes are only allowed to go. So suppose I took orange. Suppose I took orange over here. Similarly from this, either the green can come or the blue can come. Suppose once the green came. Once the green came. So whatever traits were there in the green and whatever traits were there in the orange one, that are going to combine together. That's how you get new combinations out there. Suppose another gamete was being found, again the orange one went, and from here you got the blue one. From here you got the blue one. So now what will happen? You got different traits. You are getting different traits. So this was what Mendel said. Mendel said that the alleles over here, the alleles of one pair of the gene, this is one pair of the gene, suppose capital T, small t. Okay, and this is capital R, small r. So this is for tallness, this is for the shape. Fine. So pair of allele for one particular gene are going to assort independently of the other pair of alleles for the next gene. So capital T can go with capital R, it can also go with small r. It is independently going. None of them are teaming up together, which is why you find new combinations. So in Mendel crosses, recombinations occurs due to what? Independent assortment. Due to independent assortment of genes on different chromosome. They are independently moving around into a gamete. Now my dear children, if at all you talk about Morgan's experiment, what did he do? In his experiments, basically he found that suppose there is capital A gene over here and there is capital B gene over here. Similarly, small a allele and you have small b. This is how it looks. Now, this is the parental combination of things. Capital A with capital B, small a with small b, right? Now, suppose there is a crisscross. Both of them are getting exchanged. The bottom part of both of them are getting exchanged. What will happen now? Now, small a will be there, but capital B is going to remain the same. And over here, capital A is going to be there and small b is going to be there. Don't you find new combination? Here it was capital A, capital B. Here you have small a, capital B. Here you are finding what? Newer recombinations also, newer changes as well. But this is not because of independent assortment. Remember, everything is happening within the same chromosomes. There are no multiple pairs of chromosomes. These two chromosomes are there and it is being happening within them. So in Morgan's experiment, recombinations occurs due to what? Something that you call as crossing over. Crossover is happening. Okay, and it is happening between the linked genes. These genes which are there, my dear children, okay, these genes which are there, you do not call them linked genes. Suppose they are very close to each other, you will call them linked genes. So I do not want to confuse you with that. Rather, we just say it occurs due to crossing over. It occurs due to crossing over on the chromosomes that have the same gene at the same loci. So they have the same gene at the same loci. Capital A here, small a here, but they are coding for one particular character. It can be height, it can be anything. Okay. So Morgan's experiment was in their recombination was due to crossing over, whereas in Mendel's crosses, recombination is due to independent assortment. Here you talk about more than one chromosomes, but over here you just talk about a pair of chromosomes. That's all. As easiest as that. Now, if there are obviously multiple ways to explain it, but if I dig deep into that, probably you might get confused. So I am restraining myself from doing so. This would be more than enough for you to understand, I believe. Yes, a little confusing, but it helps. Cool. Let's go ahead with the next question. Question number 3. Represent schematically the result of two gene interaction by taking seed shape and seed color in pea plant. That means you are being asked about the dihybrid cross. Two gene inheritance means one gene for one character, another gene for another character. So one character that we have taken is what? Seed shape. Seed shape can be what, my dear children? It can be round or it can be wrinkled. Now seed color, it can be yellow and it can be green. Suppose I am crossing between round and yellow and wrinkled and green parents. What is a genotype? First of all, how do you represent round? Round is a dominant character. Suppose it is homozygous, pure breed. You make it capital R, capital R. Similarly, for yellow, it will be capital Y, capital Y. Now, the recessive one is going to be small r, and small y, small y. If at all you're worried how it is actually looking, so it looks like this. So, capital R over here and capital Y over here. Now from this pair, whatever comes, it will be anyway capital R only. From this pair, whatever comes, it will be capital Y only. Right? From one pair, only one can come anyway. So all the gametes which are going to be formed, half of it will be there. In the gametes, only half the number of chromosomes will be there. So capital R and capital Y is over here. Similarly, my dear children, if at all you try to make it over here, for this particular parent, whatever you do, from this pair, small r will come. From this pair, only small y would be coming. So all the gametes would be like this. All the gametes are going to be like this. There are 4 types of gametes which can be formed, but all of them same, which is why I'm just writing once. Now the gametes, when they are fused, what do you get? Gametes, when they are fused, you get capital R from here and small r from there. You get capital Y from here and small y from there. Right? So this looks like this. Capital R, small r, capital Y, small y. Now it is looking like this. This is what the F1 generation. This is your F1 generation, filial 1 generation, where all of them are showing the dominant phenotype. Physical nature is a dominant one, that is round and yellow, because this is dominant over this and this is dominant over this, which is why all of them are round and yellow, and all of them have the genotype capital R small r and capital Y small y. Now my dear children, if I want to cross them, then what do I need to do? So for that, I will take a new slide. So I have capital R small r, capital Y small y, capital R small r, capital Y small y. The gametes now look like this. Capital R small r, capital Y small y. From this, suppose capital R has come, and from here capital Y, then one gamete will be having something like this. From here capital R and here small y, then it will look like this. From here small r and here capital Y, it will look like this. From here small r and here small y, it will look like this. How many different type of gametes? 4 different type of gametes you have got. Similarly, the same type of gametes you will be getting from here also. And how do you cross it? Basically, you write the gametes like this. All of the gametes you write on both the direction, and then you cross it, this and this. So capital R, capital R, capital Y, capital Y, this and this, capital R, capital R, capital Y, small y. Such like you have to cross. So at the end, my dear children, 4 into 4, you will get 16 progenies. How many? You are going to get 16 progenies. Out of them, if at all you are being asked about how did they look, what is the phenotypic ratio? Okay, phenotypic as in physical appearance ratio. Then the ratio is 9:3:3:1. What does 9 stand for? 9 of them are going to show the parental character, dominant parental character. What was the dominant parent character? Round and yellow. One of them is going to show the recessive parent character. What was it? It was wrinkled. It was wrinkled and green. What about them, ma'am? They are going to show non-parental character. They are recombinants, my dear children. They are recombinants. So over here, round was with yellow, right? Now we will make round with green. Round with green. Change the color. Similarly, wrinkled was with green, right? So we will make wrinkled with yellow. So these are what recombinants. 6 recombinants and 9 plus 1, 10. Basically, what do you get? You get the parental trait. This is the phenotypic ratio, 9:3:3:1. Please remember, not genotypic ratio, phenotypic ratio. Cool? So, how many phenotypes do you find, if at all you are getting a question? How many phenotypes do you find in dihybrid cross? 4 phenotypes, ma'am. 4 phenotypes. How many genotypes do you find, children? How many genotypes do you find? Now, that is a question. Different type of, what do you call a genetic makeup? How many different type of unique genetic makeup do you find? Now lots of them are going to be repeated. Okay, just similar to this, you will get a lot of other things also, but I am talking about unique. Count them only once. How many unique genotypes are possible? Remember, it is 9. So 4 phenotypes and 9 genotypes. 4 phenotypes and 9. 9 you can remember from here. The first number over here is the number of genotypes. You can remember like that. This is just for you to remember. Okay. So what do you have to do? You have to do the whole cross. I just explained it to you. This is how it exactly looks. Do not forget the ratio. Cool? Fine. The next question that we have is a 5-marker question. And this was also a 5-marker question. Yes. So question number 36. Mendelian disorders may be autosomal or sex-linked. What do you mean by autosomal? For example, in our body, my dear children, we have 46 chromosomes, right? We have 46 chromosomes. Out of the 46 chromosomes, 44 of them are autosomes. That means they are responsible for a general body character. And there are 2 chromosomes. It can be either XX or XY. If it is XX, it's a female. If it is XY, it's a male. These 2 chromosomes are sex chromosomes, which is why we say this is the 45th and this is the 46th. After 44 autosomes, we have 45th and 46th sex chromosomes. Now, if you talk about pairs, you say that chromosomes are in pairs. So, how many pairs? 46 in total. That means we have 23 pair. 23 into 2, 46, right? 23 pair is there. Now, last pair is the sex chromosome pair. So, we say 22 pairs are autosomes and 23rd pair which is there, it is what? Sex chromosome. Now, there are some disorders, Mendelian disorders, which happens when in the chromosome, suppose there is a mutation in one particular gene. One particular gene is mutated, which is why I am getting a disease, because that gene was responsible for production of something and things like that. So if at all I am getting a disease because a gene on my autosome is getting mutated, I will call it autosomal disorder. Now suppose the gene which is getting mutated is on the sex chromosomes. The gene which is getting mutated is on the sex chromosome. I will say it is a sex-linked disorder. I will say it's a sex-linked disorder. Okay, if it is on autosome, autosomal disorder. If it is on the sex chromosome, sex-linked disorder. Okay, and these are coming under Mendelian disorders. Mendelian disorders are the ones where if at all you have some mutations in the gene, you call them, and it causes a problem, you call them Mendelian disorders. Now, it can be on any gene. It can be present on the sex chromosome or on the autosomes. Now, over here, maybe autosomal or sex-linked, dominant, recessive, homozygous, heterozygous. Homozygous means same. Heterozygous means different alleles. Anyways, pedigree symbols are used to analyze these disorders in human population. Mention the genotypes of the following pedigree symbols. So over here, I have given you the answer, but you need to analyze it. So autosomal dominant disorder. Autosomal dominant disorder. And over here, you see that a square is made and the square is colored. That means it is a man, and square is for man, and color shows that this particular person has the disease. What disease? Autosomal dominant disease. If autosomal dominant is there, my dear children, you can take anything where one allele should be dominant, at least one allele should be dominant, which is why the person is going to have the disease. Now both the alleles can also be dominant. Both the alleles can be dominant, or either one allele can be dominant. Anyway, you need a dominant allele in autosomal dominant disorders. And over here picture is also given. This and this, make sure it is matching. It is matching. Okay, so I'll give you more examples, you'll see down there. So anyway, this is autosomal dominant, and here the man is diseased. That means this person can be capital A small a or capital A only. Okay, now autosomal recessive. Autosomal recessive means only in this condition they will be having the disease. Suppose they are like this, my dear children, it's a dominant thing. Both the alleles are dominant, that person will not be having disease. Both the smaller alleles, the recessive alleles should come together, then only this person will be having disease. So autosomal recessive, ma'am, small a small me. Array, see over here, in this picture a female is given and the female is not colored. That means even though autosomal recessive disorder is running in the family, this female does not have it. This female is not going to have it. So if she does not have it, my dear children, what should be her genotype? That is the question. So again, it can be either this or this. It can be either this or this. She should not have it, right? So again, the answer would be either this or this. If at all she was colored, that means you had the right small a. Recessive condition. Got it? I know it's kind of confusing, but you'll get it. Now, sex-linked dominant. So sex-linked dominant means it's on the X chromosome, right? Now, sex-linked dominant disorder will be coming if I have a dominant allele over here. Suppose capital A is dominant. So this is the condition, and this is a man, right? This is a man. So XY. And I have capital A, then this particular person will be having the disease. Will be having the disease. But over here, the person is free of the disease. That means he should not be having this allele, rather he should be having the recessive allele. He should be having what? The recessive allele. So sex-linked dominant disorder is there, but this guy does not have it. So in that case, you have to make the opposite, and that will be this. Now sex-linked recessive. Okay, sex-linked recessive. And both of them are having the disease. Both male and female are having the disease. Try to solve it by yourself. Sex-linked recessive means, my dear children, if it is a male, even if he has a X chromosome carrying the recessive allele, he will be having the disease because no one is there to overshadow the effect. Okay, he will be definitely having the disease. For a female to have it, in this condition, she will not have it. Only if one allele is recessive, she cannot have it. Both the alleles have to be recessive for her to be affected. That is why, and over here both of them are affected. You're talking about the affected people. So this is for male and this would be for female. If at all it was not, if at all it was plain, that means they were not affected. In that particular condition, over here you could have written capital A. Over here also, if you have put small a once, you should be putting capital A over here. Get it? You should not try to make it recessive. But since that is not the condition, which is why your answer would be this. Pause the video, go through it once more. I know it's not so easy to grab it in all at once, but yes, I was surprised to see these type of questions coming in. They're making you think really hard. So my dear children, yes, here it goes. Now the next question that we have, now this is from model question paper 2, question number 4. What is the possible blood group of an offspring when both parents have AB blood group? When both parents, my dear children, now you tell me the answer for this. AB blood group. That means what is the gene like? This is IA, IB, IA and IB. Similarly here also IA, IB. This and this comes together, I get IA, IA. That means the blood group will be A. This and this comes together. Ayo, just a second. Yes, this and this comes together again. IA, IB. Here A and B both are there, they show codominance. Remember, A, B blood group are example of codominance, they are equally dominant. So I got a new blood group, that is AB. Now this and this comes together, I get IA and B again. So this will be AB. this and this comes together, it will be IB, there is no one to dominate. Basically, it will be just B blood group. So I get A blood group, I get AB blood group, and I get B blood group. I get A blood group, I get AB blood group, and I get B blood group. What will be my answer? This. You are asked what is the possible blood group of the offsprings. So offsprings can have either of them. So you're not, wait. Okay, I think that was a typo. So this is not the answer, this is the answer. Okay, it's B. It is B. Next question. Differentiate between linkage and recombination. This is the 2-marker question. What do you mean by linkage and recombination? My dear children, I already explained you recombination part of it. Now I'll talk about a bit more about linkage. So I'll give you 2 scenarios, okay? Suppose capital A allele is here, capital B is here, small a is here, small b is here. This and this part got exchanged. What will happen? You get new combination which looks like this. Small a over here, but capital B still there. And small b still here, but capital A over here. You find new combinations. You find what? New combination. That means recombination happening. What do you call these type of genes as? They are loosely linked genes. What are they? They are loosely linked genes. That means they are far away from each other. Always remember, loosely linked genes will be showing high amount of recombination. New combinations will be more there. Now on the other hand, suppose over here I have capital A and the capital B gene is right over here. Similarly, small a and small b gene is right over here. Now if at all crisscross thing happens, what will happen, my dear children? Small a, small b comes together, capital A, capital B also comes together. Here do you find new combinations being formed? Very less. The upper part is different, I agree, but here between them you find lesser recombination. These 2 characters are always going together, right? What do you call these type of genes? You call them as tightly linked genes. Tightly linked genes. And my dear children, tightly linked genes will be having less amount of recombination. Will be having less amount of recombination. Now this is for your understanding. So linkage is what? It is a tendency of genes located close together on the same chromosome to be inherited together. Linkage means what? It is a tendency of the genes located close together to move basically together, to be inherited together. Whereas recombination is opposite. It is the process by which new combinations of genes are formed due to crossing over. Now over here, it reduces the chance of new gene combinations and maintains parental character. Whatever parental character was there, that is going to be maintained. But in recombination, it increases the genetic variation by producing new gene combinations out there. That is why we all do not exactly look like our parents. We also have some things happening up there. There are recombinations that are taking up in our body. So that is linkage and recombination. And who put down these terms? Who put down the terms linkage and recombination, my dear children? It was none other than Thomas Hunt Morgan. Thomas Hunt Morgan, he was working on a chromosomal theory of inheritance, and while he was working, he got to understand about linkage and recombination. Okay? And his student was there whose name is Alfred Sturtevant. Whose name is Alfred Sturtevant. Now Alfred Sturtevant, my dear children, went ahead and gave one more very important thing. I'll tell you here itself. It's known as gene mapping. So ma'am, if you tell me the percentage of recombination, the amount of recombination, by that I get to understand whether they are far or they are close. Okay, if more recombination, that means they are far away. And with that information, I can locate the genes. I can tell you the genes, where are they located. This is known as gene mapping. And that was put down by Alfred Sturtevant, who was the student of Thomas Hunt Morgan. Okay, I always say, so the next time you guys are my students, so I'm expecting something very big from you. I know I am not a good teacher like Thomas Hunt Morgan, but still I am expecting, but it's for your own good, right? So yeah. Now question number 7, schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as an example. Now this is a 5-marker question. You have to talk about incomplete dominance, and where do you see it? In Snapdragon. So what do you mean by incomplete dominance? When? Okay, I'll give you an example and I'll tell you. So my dear children, I am crossing the red flowered plant of Snapdragon with the white one. Red one is denoted by capital R, which means it is dominant. Yes, capital letter means dominant, right? And over here small r, which means it is recessive. this and this comes together, I get capital R small r, this and this capital R small r, this and this capital R small r, this and this capital R small r. All of them have only one dominant allele, and that is going to dominate. And when they dominate, what do you expect? The color to be red. But over here, what is the color that you get? It is pink. So what do you understand from that? That, ma'am, it is incompletely dominant. It is not completely dominant over the small r. Okay, it is what? Incompletely dominant. This is what exactly is. Okay, incomplete dominance. Now, my dear children, he was very curious, which is why he crossed the pink flowers and saw that what is going to happen. Let's see. So this and this crossed, capital R, that means it is going to be red. There's no small r, right? Only capital R, that is red. Now, this and this, I get capital R, small r. This is going to be pink in color. Agreed? This and this, again capital R, small r, this is also going to be pink in color, ma'am. This and this small r, this is going to be white in color. Now I see the parental characters also, but I still see incomplete dominance. That means when these two alleles come together, R is not able to be completely dominant over small r. Now, what is the phenotypic ratio over here? Physical appearance ratio, one is red, two is pink, and one is white. So what is the phenotypic ratio? Phenotypic ratio is 1:2:1, and whenever you write this, always write what do you mean by that. So 1 red, 2 pink, and 1 white. 1:2:1. Cool. Now, my dear children, what is the genotypic ratio? Genetically, how they look. So one is capital R, one, only one. Two of them are capital R, small r. And again, one of them is going to be small r. So genotypic ratio and phenotypic ratio is going to be the same. In this particular case of incomplete dominance in F2 generation. In F1 generation, there is no ratio as such, everyone is the same only, right? In F2 generation, both the ratios are the same. This is not like how Mendel studied, like when he crossed tall and dwarf plants, just like this, he didn't get this ratio. He was not getting this ratio. Genotypic ratio was 1:2:1, but phenotypic ratio was 3:1. So please know the difference. I think we have a question on that. We will be solving it as well. So this is a unique thing about incomplete dominance. Do not forget it. You also call snapdragon as antirrhinum plant. Okay? Antirrhinum majus is the name. Now there is another 5-marker question, and this is from MQP2, question number 42. Identify the genetic disorders given below. Okay, you have to identify the genetic disorders. It's a sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females, leading to defect in the cone of the eye. So it's something related with eye, and the cone is basically responsible for your colors and everything. I'll directly jump up with a solution, and that is what? Color blindness. In color blindness, my dear children, males are the one who are mostly affected. Why? Because it's a sex-linked recessive disorder. Remember, in our NCERT, only 2 sex-linked recessive disorders are given. No dominant disorders are given. Only 2 sex-linked recessive disorder, and rest of them are autosomal. Sex-linked means it is on the chromosome. It's on the sex chromosomes. The mutation is happening on the sex chromosome, right? Now, over here, what is the case that in the case of female, suppose one gene is affected. It's a recessive disorder. Do you think the female will be affected? No. Both the genes has to be affected, right? In this case, she will be colorblind. In the case of males, if this is affected, my dear children, still that person, if only one gene is affected, still that person, one chromosome is affected, still that person will be having the disease. Why? Because there is only Y chromosome. There is no other X chromosome to overshadow the effect. Which is why, if males get this particular X chromosome from any female, suppose I have a normal father. Normal father is there who do not have any problem. My mother is a carrier of color blindness. She is a carrier. She does not have this disease. When this chromosome and this chromosome comes together, the condition is like this. The male is going to be affected. the male is definitely going to be affected. But whereas in the case of females, this and this is going to come together. One normal X is there, another chromosome is affected. This will overshadow the effect. The female will be fine, which is why we say in sex-linked recessive disorders, it is the males who are mostly affected than the females. So males 8%, females only 0.4%, my dear children. And this is a defect in cone of the eye, so that is why color blindness. Otherwise, we could have also thought about what the hemophilia disease, but anyway, that is not going to satisfy over here. Next, X-linked recessive disorder affecting a single protein involved in the clotting of blood. Now, clotting of blood is related to what? A disease that is hemophilia. Ma'am, how do you say it? What happens in hemophilia, ma'am? So, in hemophilia, my dear children, one particular protein which is a part of the cascade or group of proteins which are responsible for clotting of blood, that particular gene, okay, that gene which is responsible for protein production, it is getting mutated. Now, if there is no protein, that means clotting is not going to happen properly. If clotting is not going to happen properly, if we get a small wound, the blood is continuously going to go. The blood is going to go around. So this is what the disease, and that is hemophilia, and it's a sex-linked recessive disorder. Here also the males are the one who are mostly affected rather than the females. Okay, and it's a life-threatening disorder. Color blindness, we can live with it, but here we cannot live with it. So if at all a male has hemophilia, he might not be able to live more. And if male who is having hemophilia is not able to live more, that means there is no point of mating. He cannot mate with another person because he would not be alive until then. Suppose he is mating with the carrier female also, my dear children. Suppose he is mating with the carrier female. Do you think this can happen? This and this can come together. He can give rise to a female who is having color blindness. But do you think until mating phase he would be alive? No. Which is why we say it's the males basically who are affected who die, and they usually do not pass it to the females. Why? Because they are not alive. That's the reason. If females also get it, they also would not be alive. Okay, they also would not be alive. But females have I told you it's a recessive disorder. If you have a normal X chromosome, you can still be safe. So yes, so males are the one who are affected here. Now, inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. So 3 characters are given and symptoms are given, and you are being said what is the disease. So inborn error of metabolism, it is there with us when we are born. Okay, and it is autosomal recessive disorder. That means it's on the autosomal chromosome, and it will be only coming if both the alleles are same in the recessive condition. Accumulation of phenylalanine is also happening over here. It is phenylketonuria. It is what? Phenylketonuria. What is this disorder, my dear children? So in our body, on the autosomes, there is a gene that codes for the enzyme phenylalanine hydroxylase. Okay, phenylalanine hydroxylase. This enzyme is responsible for conversion of phenylalanine, an amino acid, to what? Phenylalanine to what, my dear children? To tyrosine. Now, if this enzyme is not there, the conversion is not going to happen. If this conversion is not going to happen, this is going to get accumulated in the form of phenylpyruvic acid. And if it is getting accumulated in our body in the form of phenylpyruvic acid, what is going to happen? We might have mental retardation, and this is not even absorbed properly by kidneys, which is why it is excreted through our urine, and which is why the people who have this disease have a foul odor in their urine, will be having a foul odor in their urine as well. Got it? And my dear children, so yes, so this is the thing, and mental retardation I already told you. One more very important thing, this particular disorder is also an example of pleiotropy. Since anyway we are studying, let's study about pleiotropy also. Pleiotropy means, usually we have studied one gene, one character. One gene is responsible for one character. But over here, one gene is responsible for multiple characters. Just give me a quick second. one gene is going to affect two characters. So ma'am, only one character is being affected, pyruvic acid is being accumulated, and that's why mental retardation is happening. No baba, when tyrosine is not being produced, tyrosine is actually an important component for the formation of melanin, which is responsible for our skin pigmentation, our hair pigmentation. Now if melanin is not being produced, we will be having reduced pigmentation. You also call it hypopigmentation, reduced pigmentation. So two character at once is being affected because one gene is getting mutated, which is why it is an example of pleiotropy, not polygyny. Polygyny is something different. Pleiotropy means one gene, multiple effects. Okay, so this anyway is phenylketonuria, and now we have the next disorder, autosome linked recessive blood disease, substitution of amino acid in the beta globin chain of hemoglobin. And this is happening because of what? Some mutation in the beta globin chain. Okay, fine. The next disorder is autosome linked recessive blood disease, reduced rate of synthesis of globin chain is happening. So there is some mutation happening in the beta globin chain, and this is also there. What do you mean by this, ma'am? So I will explain you this question in the form of a long answer, because this question has come in the form of a long answer before, and I really want to explain you that right now for a better understanding. Okay, so those two diseases will be discussed over here. The question says few autosome linked recessive gene blood diseases, few autosome linked, okay, it's autosome linked gene blood disorders in human population. Among them, some are related to qualitative and quantitative problem of synthesis of proteins. So I'll tell you, in our body, you know RBC is there. RBC, my dear children, will be having hemoglobin. Will be having hemoglobin. Agreed? This hemoglobin which is there, it is made up of alpha and beta globin chains. Alpha and beta globin chains. You have a disease which is known as sickle cell anemia. What happens in this disease is that this beta gene which is there, beta globin gene which is there, it has two copies, one over here, one over here. Okay, this is being mutated. There are some changes over here happening in the gene, which is why it is getting mutated, which is my dear children, what will happen? Beta globin chain is produced, but there is a difference in the structure. In the structure of the beta globin chain, there is a difference. Okay, and with this different beta globin chain, if at all I try to make hemoglobin, obviously that structure will also be not great. Hemoglobin's function is to carry what? Oxygen. It would not be able to transport oxygen efficiently, which is why the RBC, where the hemoglobin molecule is there, it will turn into a shape of sickle cell. It will turn into a shape of sickle cell. So here, it's because of the quality of beta globin chain. It's caused because of the quality of beta globin chain which is getting hampered. Right? What exactly happens over here? So my dear children, we have suppose mRNA. We have the mRNA. Okay, this mRNA is going to code for the polypeptide of beta globin chain. This is basically a part of the protein, a type of the protein, or what do you call a basic unit of the protein, polypeptide chain you say. So mRNA is going to code for this. It has multiple codons. It will be having multiple codons, and codons basically come in the form of triplets. They come in the form of triplets, right? Now here you usually have what? GAG. And GAG, on the 6th position, my dear children, GAG is responsible for the production of glutamic acid. What? It is production of glutamic acid, which is an amino acid. Now suppose here, instead of A is being replaced by something else, and that is U. A is being replaced by something else, and that is U. So it will produce the same amino acid? No. It is going to produce another amino acid, that is valine. And that brings the whole change. Now the beta globin chain is going to be produced, but with a valine in it. And that is going to bring about the structural changes in the beta globin, and obviously changes over here and changes in the RBC as well. Okay, so it is a qualitative disorder. Sickle cell anemia is a qualitative disorder that is happening. And remember, it was A first and then U. So A comes before U, right? So you can remember A was there, it was replaced by U. Similarly, G comes before V. So it was glutamic acid first, now it is valine. So over here, mutation in the HBB gene, that is the gene coding for the beta globin chain, at chromosome number 11, so the gene is on chromosome number 11, my dear children, causes the production of abnormal hemoglobin. You say that this is HBS. This defective hemoglobin leads to sickle-shaped red blood cells, impairing its oxygen transport and causing blockages in the blood vessels. So when the shape is there, my dear children, it won't be, initially it was not able to transport oxygen because of its shape, because the hemoglobin was not oriented first. Then the cell shape also becomes disoriented into sickle shape. Now it is even more worst. It is not able to transport oxygen at all. And this my dear children, this shape of the cell is going to cause blockage in the blood vessels. Here the defect is caused because of the error in the quality of the protein which is being made. The quality of this particular protein which is being made, or the polypeptide. Protein basically is made up of what? So many amino acids. When few amino acids comes together, you call this as peptide. Okay, you call this as peptide sequence or polypeptide sequence. Now my dear children, if big polypeptides are there, you now call this as proteins. There is obviously some criteria to be called as protein, but in general also sometimes we say it's protein, or we can also say it's a polypeptide, smaller unit of protein. Now we have thalassemia, which is the next disorder. But before going there, let's go back onto the question and see if we are able to answer it now. Autosomal ring recessive blood disorders where substitution of amino acid in the beta globin chain of hemoglobin is happening. Substitution is happening, my dear children, in the beta globin chain. Yes. So what is this? This is sickle cell anemia. Now the next is autosomal link disorder where reduced rate of synthesis of globin chain is happening. Okay. Now let's go back there. And see what is happening over here in thalassemia. So in thalassemia, this was the chromosome number 11. Now this is chromosome number 16. Okay, here you have in total of 4 genes, 4 of them, which helps in the making of alpha globin chain. Here you knew, already we discussed, there are only 2, and that is responsible production of beta globin chain. Suppose one gene is being mutated over here. What will happen? 1, 2, 3 are still left to be producing alpha globin chain. But the amount of alpha globin chain which is going to be produced is going to be less. Suppose 2 of them are getting mutated, it is going to be even more lesser. Suppose all 4 of them, then alpha globin chain is not going to be there at all. If alpha is not there, can you make hemoglobin? No. Similarly, in beta chain, 1 is getting mutated, you still have 1. So beta globin chain is being produced, but the amount is what? Less when 1 is being mutated. When both, then you do not have it at all. So this disease happens because of the quantity, not because of the quality, but because of the quantity of the beta and the alpha globin chains which are being produced, which is why this is a quantitative disorder. What is this disorder? This is thalassemia. So it is caused by mutations in the genes responsible for hemoglobin synthesis, HbA1 and HbA2. So over here, suppose this is HbA1, this gene and this gene, okay, and this is HbA2. You will have 2 alleles for them, have 2 alleles for them also, and or HBB gene on the beta genes. Now in the HBB, so this is HBB gene in the beta genes. There can be mutation in 1 allele of a gene or both the alleles. 1 allele of a gene or both the alleles of a gene, which leads to what? Reduced or no production of specific hemoglobin chains, causing anemia and fatigue. There will be less amount of blood cells and there will be fatigue also. Hence the defect arises due to error in the quantity of the genes produced. Okay, it is arising due to quantity of the genes produced. That is why it is known as quantitative disorder. Now over here, autosomal linked recessive blood disorder having reduced rate. That means quantity is reduced. Rate of synthesis of globin chain will be what? Thalassemia. It is going to be thalassemia. Okay. And we will study more about it when we go through. But anyway, this is the answer for all the questions. And I think I was able to talk about all of the disorders. At least you get an idea of what the disorders are. Now question number 9. Genes located very close to one another on chromosomes tend to be transmitted together and are called as. They are very close to each other. What do you call them as? linked genes. They are linked genes. If at all we want to be specific, then tightly linked genes or closely linked genes. This is MQP3 question number 4. Now again a 2 marker, identify the symbols used in pedigree analysis. If this symbol is there where 2 lines, male and female and 2 lines is there, that means mating is happening between relatives. That is why 2 lines are being mentioned over here. And here what is happening? Male and female is mating, you get a boy child and that boy child is getting affected with the disease. So how do you write it? Parents with male child affected with disease. Parents with male child affected with disease, that is what you can write. Or basically you can say that this is a recessive disorder. It is a recessive disorder. So in recessive disorder, my dear children, probably, suppose, I'm taking like this, okay? Okay, this is a cross between XY, normal male and a normal female, but this female is a carrier. Now when this and this comes together, this female will pass the disease to the son. So it can be like this also. It's X-linked recessive disorder. It can be anything, but I'm just telling you, I'm giving you examples. Think like that. Okay, start thinking like that if at all you're not thinking. So parents with male child affected with disease is what being shown over here. Or you can also write, it can be the case where it is a sex-linked recessive disorder where the male is normal whereas the female is a carrier. Okay, going ahead. Now there are chances over here the female can give rise to normal children also. When this and this comes together, it will be normal child. Okay, represent schematically the result of one gene interaction between pure tall and pure dwarf plant. Now this is what we said, I told you in Mendel's case, we discussed about incomplete dominance, right? Similarly over here also we are taking tall plant and we are taking dwarf plant and they are pure. Pure means they are homozygous in condition. That means capital T and small t. When this and this are fused, you get capital T small t. When this and this are fused, you get capital T small t. This and this capital T small t. This and this capital T small t. Okay. All of them are going to be tall in the first generation. Now you have to make gametes properly. So I have not made it here, but I'll tell you how to make it. So you say this is the parents. Explain, parents. Now gametes means you have to put them in the circles. And then you have to do this crossing. I did it, but you have to do that. Suppose we'll do it over here, okay? Now we are crossing between them. So we have capital T small t, capital T small t. These are what? These are the parents. Now, gametes, capital T, small t, capital T, small t. This and this fuses, you get capital T, capital T. This and this fuses, you get capital T, small t. This and this again capital T, small t. This and this small t, small t. Over here, you get tall. Over here, this is also tall. Over here, this is also tall. But this one, my dear children, is dwarf. What is the phenotypic ratio? Physical appearance ratio. 3 of them are tall. Okay, PR, physical ratio or phenotypic ratio is what? 3:1, where 3 stands for what? Tall, and 1 is to what? This is dwarf. Okay, now we are talking about, this is what? Phenotypic ratio. Genotypic ratio, my dear children. Genotypic ratio, 1 capital T, 1 capital T, 2 capital T and small t, and 1 small t, small t. So this is capital T, capital T, capital T, small t, and small t, small t. This is what? The genotypic ratio. Cool? Works? Okay, I hope you got it. Here, if you see, in the case of incomplete dominance, it was 1:3:1, 1:2:1 in phenotypic ratio also. But in this case, it's different. So that's how you do it. And now, 1:5, okay. Okay. So there is one more question for 5 marker. Categorize the following as either autosome or sex-linked disorders: hemophilia, thalassemia, phenylketonuria, myotonic dystrophy. So hemophilia is what? Sex-linked disorder, sex-linked recessive disorder, right? Thalassemia and phenylketonuria, both of them are autosomal. Remember, in your NCERT, 2 diseases sex-linked, color blindness and hemophilia. 3 diseases autosomal linked, and that is what, and recessive also, that is sickle cell anemia, phenylketonuria and thalassemia. Okay, so these 2 are autosome, this is sex-linked. What about myotonic dystrophy? Ma'am, it's not there in our NCERT, it's there. In the pedigree picture, they are given the example of myotonic dystrophy also. And this is the only example for Autosomal dominant disorder. This is what? Autosomal dominant disorder. That means, suppose it's like this. This person will be having the disorder because he has what? One capital allele. Okay, dominant allele are there. So your answer for this question becomes what? Yeah. So that is 3 of them autosomal and 1 of them sex-linked, but I have in my answer categorized properly. So it is X-linked recessive, this is autosomal recessive, this is autosomal dominant. You can also do that. It gives you extra goodies. Now, what conclusion will you arrive after careful observation of the below given diagram? So below given diagram is what, my dear children, over here, valine, histidine, leucine, threonine, proline, valine, glutamic acid is given. At the 6th position, if you see 6th position, valine is being formed because GUG is there. That means there is a mutation that have taken place, and this is what you see in sickle cell anemia. Yeah, this is what you see in sickle cell anemia, where GAG, A, is being mutated to what? U, or is replaced by U. So instead of forming glutamic acid, now you have valine being formed at the 6th position. So from the diagram we conclude that a single base substitution, point mutation has taken place. What do you call this? Point mutation. At one single point, instead of GAG, it is GUG now. From the diagram we conclude a single base substitution in the mRNA codon has occurred. The codon becomes GUG, which codes for valine. So in the beta globin chain, glutamic acid at the 6th position is replaced by valine, a point mutation leading to sickle cell anemia. Now next question, identify the disorder by the following symptoms. Individuals with overall masculine development with gynecomastia. So they have the overall masculine development, but they have gynecomastia. What do you mean by that? Development of breast, that is what you mean by that. So overall masculine feature is found only in the case of male, but if at all you have some female character, that means probably you have one extra X chromosome. And where is this condition where you have extra X chromosomes? So 44 autosomes are there, but instead of having 2 sex chromosomes, you also have 1 more. This is the case in the case of Klinefelter syndrome. Klinefelter syndrome. Okay, Klinefelter syndrome, 1 extra X chromosome is there. Is it a Mendelian disorder? No. Mendelian disorder mean in 1 chromosome a gene is being affected. That is what you mean. Here extra chromosome altogether is coming. Okay. So it's a chromosomal disorder. It will be chromosomal disorder, and your answer for this question is Klinefelter syndrome. Got it? Yes, ma'am. Now there is one more question, and that is explain thalassemia as an example of Mendelian disorder. Try to solve it and I'll be right back. Okay, so we are back with thalassemia. So it says that it's an autosomal recessive blood disorder. Ma'am, we already know that. Okay, it is transmitted from parents to offspring when both the parents are heterozygous carriers. That I'm sure you do not know. So my dear children, for both the parents to be heterozygous carriers, what do you mean by that? That means one allele, suppose it's like this and other one is like this. Similarly, one is like this, the other one is like this. So this is the recessive allele, agreed, and this is the dominant allele. They are heterozygous because both the alleles are different over here. Now suppose, my dear children, this and this comes together. You get HBA, HBA, both of them small. In this condition, my dear children, don't you think the person will be having the disease? Yes. But the parents, they are carriers because only one is the affected allele. The other one is the normal allele. Here also one is affected, the other one is normal. But in this case, when the carriers come together, they can give rise to progenies having the disease, which is why we say you should take genetic checkup being done before, before planning for a baby. Right? Why? Because there are cases where you and me, everyone might seem that we do not have any disorders, but we might be carriers for it and we can transmit it to our children, our future generation out there. So the disorder occurs due to mutation or deletion of genes involved in hemoglobin synthesis, leading to a reduced rate of synthesis of alpha or beta globin chain. Alpha globin chain HbA1, HbA2, I told you there are 2 genes having 2 alleles separately, are located on what? 16th chromosome. Remember, 16th chromosome, not on 11th. Questions can come. Mutation or deletion of one or more of these genes reduces alpha globin production. Similarly, beta globin gene is located on 11th chromosome and mutation in one or both the alleles affects what? Beta globin synthesis. Reduced globin synthesis leads to abnormal hemoglobin formation and anemia, which is a characteristics of thalassemia. This is the gene I was talking about. So for alpha, you have 2 genes with 2 alleles. HbA1, HbA2. Here you just have 1 gene with 2 alleles, that's all. Okay? Now, this is a homework question for you guys. Which three scientists? So this is actually not model question paper 2, rather this is model question paper 3. Okay, I think somehow I missed it. I mistakenly put it as 2, but this is 3. This is for visually challenged children. This question was for visually challenged children. So 42nd question was this only. Okay, this question that I showed you right over here, this was a 42nd question, and now for visually challenged children you had this question. This is why I explained both of them. Fine. Now homework question, which three scientists independently rediscovered Mendel's work? Avery, MacLeod, McCarthy, Sutton, Morgan and Bridges, Bateson, Punnett and Bridges, De Vries, Correns and Schemark. You can let me the answer. You can let me know the answers. Okay. Now, the revised question bank questions as well as the previous year MQP questions or anything else which I feel is important, I put over here. Let's discuss them first. So, plant having the genotype capital A, small a, capital B, small b will produce dash kind of gametes. How many type of gametes? So, always remember there's a formula and that is 2 raised to the power n. Okay, over here n stands for number of heterozygotes. Number of heterozygotes, my dear children. So here this is a heterozygote number 1, this is another heterozygote, this is 2, right? We have 2 heterozygotes. Why do I call it heterozygote? One is capital A, small a. If it is capital A itself, you call this homozygous condition, but now here it is heterozygous condition. Fine. So number of heterozygotes, ma'am, we have 2. So 2 raised to the power 2 is equals to what? You have 4 gametes going to be formed. You would be having 4 gametes which are going to be formed right over here. Next question, which of the following is not a pair of opposing characters Mendel studied? So Mendel studied 7 pairs of contrasting characters. 7 pairs means 14 traits all at once, my dear children. So what are, you have to tell among them which is that pair which is not the character that Mendel studied. So axial and terminal flowers, did he study that? Yes. Axial means, suppose this is a stem, from a place where bud is coming, from there my dear children flower is coming. This is axial. Terminal means at the tip flower is coming. Which is dominant? Axial is dominant. A comes first, so axial is dominant. You remember like that. Okay, so this is a thing that he studied. Full unconstricted pods. You might have seen green pea comes in pods. We see green pods, yellow pods, right? So which is dominant? The green one. We pick up the green one, right? So green is dominant. Oh, sorry, we'll come to color. Basically, full is dominant and constricted is recessive. Constricted means it's dry. Full means it's full flattened up. We think that it's nice pea, right? So full and constricted. So this is there. Green and yellow pods. Yes, green color is dominant in pods, and yellow will be recessive. If at all you talk about the seed color, my dear children, even though we eat green color peas, but actually yellow is the one which is dominant. So that is why we have studied round and yellow and wrinkled and green. So yellow is the dominant. You remember that. Don't get confused between pod color. It's opposite there. Now pink and white flowers. I have not studied it. Okay, so it's not pink, it's actually violet and white flowers. Violet is a dominant character, white is a recessive character. So this is wrong. Cool? Yes, ma'am. So these are the traits that he studied. Please go through. For flower, you have 2, flower position and flower color. For pod shape, you have 2, pod shape and color. And seed color and seed shape is there. One thing is unique and that is stem height. Chromosome maps or genetic maps were first prepared by genetic maps, gene mapping. Sutton and Bovary, Bateson and Punnett, no. Morgan, student of Morgan, his name is Alfred Sturtevant. I told you, right? So your answer for this question would be D. Next, which of the following is incorrect regarding ZWZZ type of sex determination? Remember, the different types of sex determination are super important. Okay, you have to study it. So ZWZZ type. ZW and ZZ type of sex determination, where do you find it? You find it in birds. And birds are unique because in birds you find something that you do not find anywhere else. And that is female heterogamety. Female heterogamety means females are going to produce 2 type of gametes, either Z or W. Males are going to produce 1 type of gametes, they are going to be just Z. Suppose this and this fuses, you get ZZ, again a male. This and this ZW, that is going to be a female, and that is what you see in birds. That is what you see in birds. So you are being asked, so none of the other organisms, for example, if we take human beings has XX, XY type of sex determination. Over here, males are producing 2 type of gametes. Males are heterogametic, 2 type of gametes, but females are not. If you take Drosophila also shows this only. So humans and Drosophila have XY sex determination. Both of us, my dear children, have male heterogamity. Now there is insect. In insect, my dear children, it is XO. Females are XX, males are XO. XO means there is one chromosome, there is a lack of one chromosome. So in this condition, it is going to be male. Suppose here I got X chromosome, but from here I didn't get any chromosome. Okay, the gamete is khali of the chromosome. In this case, it will be a male. Two type of gametes are produced by males, right? So this is also coming under male heterogamety. Two cases, male heterogamety, only one case, female heterogamety, and that is in birds. This is in insects. This is an insect, example grasshopper. This is in human beings and also Drosophila. Drosophila also comes under arthropods, but somehow they are being moved away. So it occurs in birds. It is not incorrect. You have to find what is incorrect. So this is correct. Females are homogametic and males are heterogametic. Are females homogametic? Homogametic means same type of gametes are being produced by the females. Wrong. This is the incorrect statement. Females are heterogametic and males are homogametic. Correct. Total number of chromosomes is same in both male and female. Yes, it is the same in both female and the male. This is also correct. The only incorrect statement that we have is what? I have marked it wrong. This is the answer. Okay, I have marked it wrong. This is the answer. Do not worry. Answer. I'm writing it again. Okay? Fine. A woman with normal vision whose brother is color. Okay. This type of question usually comes up, so you should be very much aware. Let's see. A woman with normal vision whose brother is colorblind. Okay. Marries. Normal vision. Woman is with normal vision can be like this. XX or probably can be like this also. She can be a carrier, she will be having normal vision. Whose brother is colorblind marries a man. Okay, she marries a man with normal vision. For a man to be having normal vision, it can be just like this. They have 4 children, 2 sons and 2 daughters. Okay, one son is colorblind. Okay, if the son is colorblind, the female cannot be like this. The female has to be like this. So let's remove this. Everyone went. Okay. One son is colorblind. That means this chromosome. So that means this chromosome and this chromosome came together. While the other son and both the daughters have normal vision. Other son, that means the one which is formed like this, and the daughters have normal vision. Yes, we'll have the pattern of inheritance for the disease is what? Sex-linked recessive, sex-linked dominant, autosomal recessive. We know it's about color blindness. It has to be sex-linked recessive disorder. It has to be sex-linked recessive disorder. To as many questions as possible from this, you might get numerical type of questions also. It's easy, trust me. You will get it only if you try to solve it. You might be asked, if you cross this, how much percentage? Let's try this. Anyway, we are solving this only. Let's try this, okay? So I have XY and I have XXC. Let's see how much percentage of boys are going to be affected. This and this I get XX. This and this I get X. This and this I get XY. This and this I get XCY. This is a male who is affected. This is a male who is safe, who is not affected. So out of 2 males, 1 is being affected. So can I say 50% of males is being affected? If the question is how much percentage of male is being affected, you have to talk in terms of males, not about in terms of all of them. So 50%. If at all you are asked in all the progenies, how much of percentage of them are having color blindness? So only 1 has color blindness out of 4. 1 by 4 is equals to what? 25%. How many females are colorblind? 0. They are not colorblind. They are carrier. They are not colorblind. 0%. Okay. Now, how much percentage of females are colorblind? Take half of it. Only 1%. Only 1 is colorblind, right? So half of it. 50% of female will be, carrier, not colorblind, carrier. Fine. So please know about these small things. It's super important. Try to solve as many questions as possible. I think I have a question. Okay. So if both, this is for thalassemia, that means autosomal recessive disorder. But anyway, let's solve it. If both parents are unaffected carriers, in thalassemia, unaffected carriers means it cannot be small a, then they will be having the disease. They have to be carrier, that means 1 is dominant. Okay. Which is an autosomal recessive disorder. Fine. What are the chances of pregnancy resulting in an affected child? So out of all you are being asked, affected child. So this and this crosses, let's see, you get HBA, normal child. This and this, you get HB capital A, HB small a, carrier child. This and this, you get HB capital A, HB normal a, normal child. I mean carrier. This and this, both of them are small. HBA, HBA. Out of 4, 1 by 4 is what? 25%. 25% of them. Okay, what are the chances of pregnancy resulting in an affected child? 25%. Got it? Cool? Yes, ma'am. Next question. Now this is a 2-marker question that has usually shown up. What is haplodiploid sex determination mechanism? Mention an animal which exhibits it. So ma'am, you told in birds you have ZW, ZZ. In human beings and Drosophila it is XX, XY. In grasshopper it is XX and X0, X0. Now you are saying there is another type of sex determination? Yes, and that is haplodiploid sex determination. By that I mean, haploid means what? Suppose, my dear children, if I say that we have 46 number of chromosomes, this is what? In a diploid condition. In diploid condition, if I talk about haploid N, it will be what? Half of it, that is 23. Suppose I am saying that an organism which is diploid in nature, now this number can be anything. Suppose I change the number here to 32, and here half of it is what? 16. So an organism having 32 number of chromosome is going to be a female, and the one having half the number of chromosome is going to be a male. If at all I say this, you have to believe me, because that exactly is happening over here in the case of honeybees. So rather than asking you what is haplodiploid sex determination, they can just ask you, explain the type of sex determination in honeybees. So this is it. So what exactly happens is, females having 32 number of chromosome. Now when you want to form gametes, you undergo meiosis, reductional division. Half of the number of chromosome is going to be there in the gametes. Here the males are already haploid, my dear children. They cannot undergo meiosis, they undergo mitosis to increase the cell number. Chromosome number remains the same. Now a gamete carrying 16 chromosome and a gamete carrying 16 chromosome comes together, you get an organism having 32 number of chromosome, it is going to turn into a female. Ma'am, how do we make males then? If no fertilization is happening, suppose this gamete formed by the female, here no fertilization is happening, still it is being converted into organism. This is known as parthenogenesis. Without fertilization, my dear children, male is being formed. Which is why we say 16 number of chromosomes are only up there. And if you notice, this son does not have a father. Yes, this son does not have a father, but definitely has a grandfather. This is a sentence in your NCERT, you need to remember it. The son does not have a father because it's just from the mother's egg cell it is being formed, but does have a grandfather. Why? Because 32 came because male and female chromosomes came together. So obviously male was there, grandfather is there, son is not there. So please remember that. So males develop from unfertilized egg and are haploid, having a single set of chromosome. That is what you mean by haploid. Females develop from fertilized eggs and are diploid, having 2 sets of chromosome. A queen bee lays egg. If the egg remains unfertilized, develops into a male drone. Yeah, so it is male drone. Males are usually just for sexual activity, nothing else. Okay, and yes, so that's all. Know about it. Super important. Now there's another question. Define point mutation. Give an example for point mutation. Point mutation means a single nucleotide pair is being changed. I told you it was GAG first, now A is replaced to U, it is GUG now. This is example of point mutation. Change in single base pair of DNA is known as point mutation. A classic example of this is mutation in sickle cell anemia, where the codon GAG, the second nucleotide, that is A, is replaced by U, hence making the codon as GUG. Note that GAG codes for glutamic acid, but GUG codes for valine, changing the whole structure of beta globin chain. So please know that. Now, what are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila with respect to linkage? So I told you he was doing experiment that is chromosomal theory of inheritance with the help of Drosophila melanogaster, and he got to understand a lot of things about linkage and recombination. So what were his understanding? The higher the linkage, okay, the higher the linkage, the lesser the recombination. That means the higher they are closely linked, the lesser recombination will be there, and the lesser the linkage, that means if they are far away, my dear children, there will be more recombination. In other words, when genes which are tightly linked will show lesser recombination, and genes which are loosely linked will show higher recombinations. As simple as that. Okay, now I'll tell you one very important thing. In his experiment, there were two characters, yellow body and white eye. White eye. These two genes were very close to each other. They were very close to each other, which is why the percentage of recombination was just 1.3. Was just what? 1.3. That much. Only that much percentage of recombination was there. On the same hand, there was wing structure and white eye. They are very far from each other, which is why the percentage of recombination was more there. It was 37 point something percentage. So that is more recombination. So please remember, yellow body and white eye. Yellow body and white eye, lesser recombination. White eye and wing color, my dear children, more recombination. Or yellow body and wing color, more recombination would be there, because they are far away from each other. Okay, now T.H. Morgan selected fruit fly for his experiment. Why? Give any three reasons. So they could be grown on simple synthetic medium in the laboratory condition itself. They didn't need any fancy stuff out there. They complete their life cycle in about 2 weeks, so which is why we can see the results within the 2 weeks itself. And single mating could produce a large number of progeny, so you can do more experiments. There was a clear differentiation of sexes. The male and the female flies are easily distinguishable. Males are usually smaller, females are the one which are larger. Okay. Now what are chromosomal disorders? Give an example. Chromosomal disorders means, I told you, if at all, suppose a chromosome number is less or more, or if at all there are some structural problems in chromosome, which is why you get a disease, you call them as chromosomal disorder. Disorder in human beings are caused due to absence or excess or abnormal arrangement of one or more chromosomes are called chromosomal disorders. 3 examples are given in your NCERT: Down syndrome, Turner syndrome, and Klinefelter's syndrome. I have one more question related to it, and that will be making you understand things a lot better. Just give me a second. Yes, this one. Define aneuploidy. Give 2 examples of aneuploidy. So aneuploidy is a reason why you get chromosomal disorder. Aneuploidy means loss or gain of a chromosome. Loss or gain of a chromosome. If at all you're gaining, you call this condition as trisomy. If at all you're losing, you call this condition as monosomy. So examples you're being asked. Okay, so let's discuss. Down syndrome. You have Down syndrome, you have Klinefelter's syndrome, and you have Turner syndrome. In Down syndrome, the 21st chromosome, it's an autosome. Okay, it's an autosome. The 21st of chromosome, it should be like this, one from father, one from mother, but you have one extra thing, which is why we say it's trisomy of 21st chromosome. So if you are asked the karyotype, how many autosomes are there? 45. We have usually 44, but here 45 autosome is there, and can be XX, then it is a Down syndrome patient who is a female. It can be 45A XY, then it is a Down syndrome patient who is a male. Okay. Now Klinefelter syndrome is allosomal disorder. That means it's not on the sex, it's not autosomal chromosome which are being increased. Rather, it is allosomal. Sex chromosomes are known as allosomes. So I'll say allosomal. So usually we should be having XY, but here one extra X is there. Again, this is a trisomy condition. So overall masculine development is there, but my dear children, they will be having gynecomastia, formation of breast, and they will be sterile. They will be sterile. There won't be sperm production. Please remember, no sperm. Okay, so they are also going to be sterile in nature. No sperm production. Spermatogenesis is not going to happen in them because of one extra X chromosome. Now Turner syndrome is where you should be actually having XX to be a female, right? But over here, other chromosome has not come. Less chromosome is there. So I'll say this is a monosomy condition. This is what? Monosomy of the X chromosome out there. This is also allosomal. Here the female is sterile. Again, ovaries are not being formed, they are rudimentary, which is why again they would be sterile in nature. Fine. So this is what you had to remember, and if at all you are asked about the karyotype of Klinefelter's, you have 44 autosomes plus XXY. you also write it as 47XXY. So you can write both of them. By 47 you means instead of 46 chromosome you have 47, and then you put XXY. You can write both of them. In Turner syndrome, my dear children, you have 44 autosomes, but this is XO condition. You can write like this, or you can write 45XO. Instead of 46 you have 45. Cool? Yes, let's see the definition for aneuploidy. Is a condition in which an organism has an abnormal number of chromosomes, either due to gain or loss of one or the more chromosome. It is caused by non-disjunction during cell division. Non-separation during what? The cell division. During cell division, it is proper separation is not happening up over here. Then you talk about the examples. Please go through these symptoms really well. Fine. You have one more thing that is known as polyploidy. Polyploidy means a total, one set, a whole set of chromosome, my dear children, its number is increased. A whole set of chromosome has been passed, not one or two, whole set. That usually happens in the case of plants. So P for plants, P for polyploidy. This does not usually happens in us. Now we go back to this question. Where is it? Okay, a simple cut results in non-stop bleeding in hemophilia affected patients. Okay, give reasons. Why? Because the protein which is responsible for clotting, my dear children, that is being mutated. The gene that codes for the protein is being mutated. So how do you write it? Non-stop bleeding occurs when a single protein that is a part of cascade of proteins involved in the clotting of the blood is being affected, or the gene is actually being affected. Now, Turner syndrome affected females are usually sterile. That means they cannot give rise to babies. Why? Because females are usually sterile. They have only one X chromosome, leading to underdeveloped ovaries and lack of normal gamete production. That is why cannot have babies. In Morgan's dihybrid cross, in Drosophila showed flies having genes for yellow body and white eyes exhibited less recombination. Why? Because they are tightly linked. They are very close to each other. I just told you they are very close to each other. Yellow body and white eyes. So yellow body and white eye genes in Drosophila show less recombination because they are located close to each other, same chromosome exhibiting genetic linkage. Now, I'll go with this and then I'll come to this, okay? Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why? Because the gene which is responsible for conversion of phenylketonuria is not being produced. It's a disorder. Phenylalanine accumulates in the body to a defect in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing what? Toxic buildup. And now, inheritance of skin color in humans shows different phenotypes. In skin color, my dear children, do you think we have different phenotypes? Yes, multiple. Why do you think we have? Because it is an example of polygyny. Remember, your skin color as well as your height is an example of polygyny. Polygyny means multiple genes are responsible for one character. So pleiotropy means one gene, multiple character. Polygyny means multiple genes are responsible for what? One character. So suppose over here, I have something like this. Capital A, capital A, capital B, capital B, capital Z, capital C. This genotype will be capital A, capital A, capital B, capital B, capital C, capital C. This is the genotype having the darkest skin tone. Suppose over here, it's a combination effect. Suppose over here, one allele is small a, then it will be capital A, small a, capital B, capital B, capital C. This is lighter skin color than this. And the lightest will be what? If all of them are recessive, that is this. In this condition, you get the lightest skin color. So what do you understand? That multiple genes are responsible for the trait. That is your skin color as well as your height. This is polygyny. Okay, questions can be coming. Which genotype has the lightest skin color? This one. Okay, it shows different phenotypes due to polygenic inheritance where multiple genes contribute to phenotype, leading to a range of different skin colors. Got it? Yes, ma'am. And I think we have completed all the very important concepts, my dear children. Please go through pedigree chart as well, and I will make sure from my end that I'll give you another video for pedigree chart. If at all you want me to make a video on that, put it down in the comment section. I'll take your consideration, and I'll take your request also into consideration, and I'll think of a long video, or probably I was thinking of doing a shorts, but if it helps, I can make. So that's all, and do not forget to hit the subscribe button. I'm reminding you again, if at all you feel that this session was helpful for you, and keep on practicing, you guys are gonna absolutely rock your PU2 boards. So bye-bye, take care.